I'll- Hello everyone. Am I audible to you? Okay. I'll start at 6:05 because I let other, if someone wants to join, they may join. We'll start at 6:05. Okay. Okay, let's start with today's session. So, hello everyone. Today is our week 8 live session of Introduction to Proteomics subject, and this is going to be our last class. I hope your exam going to be on 21st or maybe 22nd. I think it's on 21st. I'm not sure about the exact date, but all the best for your exam. So today we will be ending our session where we will be talking about application of proteomics. Okay, so today's class going to be mostly application of proteomics, and then I'll talk about targeted proteomics using triple quadrupole MS, and we'll discuss more in detail about selected reaction monitoring and parallel reaction monitoring. So talking about the application, I'll talk about the application while we will discuss these modification, or you can say these advancement of targeted proteomics. So I have already discussed about targeted proteomics. So if just to give you a brief again, targeted proteomics is when we have some knowledge, when we are not doing any kind of a discovery, rather we have a prior knowledge of what we want to study. So whenever we do a proteome analysis, we know that we have to take few sorry we have to study these many proteins. These are under our, we have to observe the expression of these many proteins. Then we do a targeted proteomics because we are aware of that we have to focus on this particular type of protein or family of protein um, specifically. So here you have a more of targeted approach, right? This is not a discovery proteomics because so sorry. In discovery proteomics, you are not aware of any prior knowledge, but it's just that you want to know the global proteome of a particular. Someone wants to say something? Okay. So in discovery proteomics, you want to understand the global proteome of a particular cell, organism, or tissue, as well as you want to see the changes. But in case of targeted proteomics, you will take some known proteins and you want to study the changes in that particular protein only. So this is what targeted proteomics is. So if I talk in details, these are methods for quantitating a limited number of known protein with a high precision and accuracy in total protein extract. So you have the prior knowledge, you want to study a limited number of protein only because there are lot many protein. And even when you do a global proteome analysis, when you want to acquire all the absolute quantification you want to do, then only you will leave few proteins which are not known or which may get lost during the sample preparation. There are lot many things, but in case of targeted protein proteomics, where you have a prior knowledge and you will specifically target your known proteins, and then at that particular time you have a set number of proteins on which you want details in terms of precision as well as accuracy. You want to check the gene expression of these many proteins only in case of targeted quantitative proteomics. So this method can particularly useful for low abundance targets. So for example, in the last class also I have told you this thing, and rather I've been telling you this thing for a very long time, that whenever we do a biomarker discovery or something that helps you understand the disease, uh, understand the development or progression of the disease, whether it's a prognostic marker or diagnostic marker, you may get these marker in low abundance targets only. So for example, if I have serum as a sample, like I have isolated serum from some cancer patient, so serum itself contains some proteins which are abundant in a particular type of sample. So serum is a sample where you will find more of albumin, such kind of a proteins are abundant in nature. So they may mask the presence of low abundant protein. So whatever changes happening at the low abundant protein or low abundant targets, that may not be captured by your technique. So to get these kind of to get the knowledge of low abundant targets, you sometimes do targeted proteomics, or you treat your sample, reduce the complexity of the sample, and then perform the whole global proteome, or you can say discovery proteome. When you don't know, when you don't have any knowledge of what all proteins that you have to study, then you treat your sample, you remove the complexity. Removing the complexity means you are removing all the abundant protein that may mask the presence of low abundant proteins, and then you proceed with mass spectroscopy. But when you come to targeted proteomics, where you are aware of that what all protein that you need to study, then you then it become very easy for you to get the information of these low abundant targets, because you have the number of proteins that you want to study. A certain amount of method development, previous data, or sample preparation is required, regardless of which these methods are used. So as I've mentioned, whenever you are doing any kind of a proteomics, you treat your sample, you make your sample in a form that it can be readable. So for example, if you not going to treat your sample in terms of their complexity, you may have a masked result, and that will not be accurate. And to increase the reproducibility as well as the to get the meaningful information from your proteome data, you have to do some pre-research in case of targeted quantitative proteomics. But even if you're doing a discovery proteomics, you need to treat your sample regardless of what method you are using, regardless of what kind of a high throughput techniques you are taking up, you have to treat your sample. So this is how it is very important. So here they have given you detail about PRM, which is parallel reaction monitoring, and likewise you have SRM. So we'll discuss this thing in detail, but for now, I think this much information is very, I mean, the things that you have to keep in mind while doing any of the proteome data is uh, understanding the kind of samples you're going to use. Second, you have to prepare, sample preparation has to be done in a very precise manner with same methods that you're going to use for your uh, next set of experiment. Because to maintain the reproducibility, you have to maintain the procedure as well as the you have to take a single procedure, and uh, as well as you have to reduce the complexity of your sample. Reducing complexity, I have already mentioned you this thing. Okay, so if I talk about the, okay, let me just use the laser pointer. So when we do a discovery MS or something, so we have lot many proteins to study. But when we have a target selection, now we are aware that we in first step is always discovery MS, where you have proteome. First MS is always contain all the protein that going to get ionized, and it will read all the protein. Now, once you have the complete proteome, then you select your target. So you have the detail of your protein that you want to study. So you select your target, and that selection, then you move to I mean detail. So when you use the data of discovery MS, then you are aware that okay, fine, these proteins are present in your sample. And then once you are aware that these proteins are present, then you take your sample and then you move to high throughput mass spectroscopy, where you're going to do your target quantitative proteomics, either using quadrupole system or any other um, hybrid technology you can also use. But depending upon whether you want to do a target study, relative quantification, absolute quantification, you make your workflow. We have talked about workflow in our previous class, so I'm not taking that into detail. But if you could see here, once that person has selected his target, now he is taking that selected selection of precursor ion which he has done from the discovery MS, and then this is sent to other mass spectroscopy to understand the details of selected precursor ion. They are getting fragmentation further because this red one is selected and it is undergoing the fragmentation process for second MS. And once it's sent to MS2, the detail of this particular protein will be uh, resolved on your spectra. Okay, so in case of your parallel, we'll talk about parallel and selective later, but in case of targeted quantification, you have the prior knowledge of what all you need to study. Likewise, you have selected reaction monitoring. This is again a similar sort of a setup where you have three quadrupole system, and each one eliminate what you don't want. So here basically ये Q1 Q2 Q3 को इस तरीके से हम सोच सकते हैं कि they are just like a gate. Okay, so you are putting a gate, you're just letting those protein go further which you want to study. That's why it's a targeted proteomics. So you have whole cell genome, you don't want information from whole cell proteome, right? You don't want all the proteins to be started, because that will actually make your data very complex. So you are selecting a particular protein, you're selecting your ions by performing first discovery proteomics, and then from there you're selecting your target, and then sending your targets to these quadrupole system, right? And this will make your data more resolved as well as you can easily understand the expression of your target multiple proteins. You don't have to study the entire proteome because that will be huge. Sometimes you even get 30,000 proteins in your one proteome data. So it will become very difficult to study each and every one, right? So when you have a target, because definitely when you do your discovery proteomic, you have to study all those proteins. But when you have your targeted proteomic analysis, then you choose your proteins, and then only those proteins will be studied further. Okay, so this is how it is. Then let's talk about what is quadrupole mass spectroscopy. So we have talked about tandem mass spectroscopy, we have talked about LCMS, we have talked about lot many things, but we have not talked about in details about we. I have given you a brief of quadrupole mass spectroscopy when we had our introduction to mass spectroscopy, but today we'll talk about it in a more detailed manner. So quadrupole mass analyzer was first developed in parallel with the quadrupole ion trap by the third Nobel Prize winning mass spectroscopy pioneer Wolfgang Paul. So he is the one who introduced this analyzer as well. So we have talked about quadrupole ion trap, where I have told you that we use four different uh, four rods, or you can say poles, which these are parallel rods. Two of them are negatively charged and two of them are positively charged, and depending upon your ion mass to charge ratio, uh, these ions pass through these four rods. They actually make their own path, right? Depending upon the kind of repulsion and attraction they are experiencing in this particular system, where you have two negatively charged parallel rods and two positively charged parallel rods. Okay, so they make their own path, and that's how each ion take its own time to reach to the detector, which is which the detector going to record. So that is how you resolve your spectra in this case. So ion produced in the source of the instrument are then focused and passed along the middle of the quadrupole rods, which are four rods, two positive, two negative. Their motion will depend upon the electric field, so that only ion of a particular mass charge ratio will be in resonance. So yes, there is an electric field, and that's why they make their own charge. So depending upon the mass to charge ratio, there will be a resonance that is generated because of presence of electric field, and thus pass through the detector and pass through and reach to the detector. So the RF is varied to bring ion of different mass to charge ratio into focus at the detector, and thus build up a mass spectra, because each mass to charge ratio have their own path. As I have already mentioned, that they will take their own path depending upon the kind of electric field they are experiencing, resonance they are experiencing, attraction and repulsion they are experiencing. The trajectory of ion through the quadrupole is actually very complex, because it seems very simple that it's moving like this, but it may move in any direction. So it's a very complex, you know, it it look like that it's going to move like a wave. No, it is a very complex path. So the figure is more simplified form. Okay, so don't go by just figure. They take a lot of difficult paths and then they reach to the detector, and that is what is uh, that is what is analyzed by further. So detector detect the path and detect the time and everything depending upon the kind of mass to charge ratio that particular ion contains. Then when we talk about in detail, the two opposite road in the quadrupole have potential of positive, जिसे हम label करते हैं as a positive, and the other two has negative. So this is what the detail I've here. I'm so sorry. So if you see here, U is the fixed potential and V cos omega t is the applied RF of amplitude V and frequency W. Just give me a second. I'll take some water and then we'll get back to you. Okay. Yeah. So, so this is basically the frequency, or you can say RF potential they have mentioned. So RF potential of the amplitude is V, which is here V cos omega t. Omega t is the frequency. So the applied potential on the opposite pair of the rod varies sinusoidally as cos omega t cycle with time. So you have seen here that it moves like in like a wave, right? Up and down, and that depend upon the cos value with time. This result in ion being able to traverse the field free region along the central axis of the rods with but with oscillation amongst the pole themselves. So if you could see here, जो axis बन रहा है, there is a generation of an axis which is in between these rods, and there is a proper wave-like movement of these ions. These oscillations result in complex ion trajectories dependent on the mass of to charge of the ion. Specific combination of the potential mu, v, and frequency omega sorry, will result in specific ion being in resonance, creating a stable trajectory through the quadrupole to the detector. So basically अगर हम ये कहें कि एक particular mass to दो ions हैं जिनका mass to charge ratio भी same है, and then we have a constant u, v, and omega, specific combination, basically omega is going to change, right? So fixed mu होगा, rest of the thing will change. So अगर हम सारे combination भी देखें तो there could be a possibility that there is a stable trajectory of two ions or similar trajectory, but at the same time all the ions going to have their own complex trajectory depending upon these parameters. So all other mass to charge values will be non-resonant and will hit the quadrupole and not be detected. These mass range and resolution of the instrument is determined by the length and diameter of the rods. So what happens? So there are some, see यहाँ पर अगर आप देखोगे, so जो ये वाले ions हैं which are hitting the rods because they did not have that, they are not. सो इमेजिन, एक बार इसको थोड़ा सा 3D में इमेजिन करके देखना कि यू हैव इलेक्ट्रिक फील्ड एंड यू हैव लॉट मेनी आयंस विद सम मास टू चार्ज रेशियो, राइट? सो some of these ions will not experience any resonance, right, depending upon their mass to charge value, and they may directly go and hit these rods. So in that case, these ions will not be detected by the detector. Okay, so that's why I always say that the proteome, whenever you are reading your proteome, it depends upon lot many things. If some of the proteins are not getting converted into ions, they are unable to get fragmented and getting any charge, they will not be detected. So in case of targeted, you have known proteins. That's why it become little easy, because then you will be reading some specific protein. So you are aware that whether your samples are being prepared with the whether your samples are prepared in a good manner or not. If you're not getting your desired protein, details of your desired protein, you may get an information that things might not work well during your sample preparation. But when you're doing a discovery proteomics, where you have no prior information, that whatever samples you are preparing, you will always have some dilemma whether things went wrong during sample preparation, or this is what I'm getting from my sample. Because when it comes to what you say clinical sample, then you have to be little more careful, because you may not get those samples again, right? So these are the things that you have to consider, because proteomics mass spectroscopy also have its limitations. So if the ions are not formed, if they have not experienced any kind of resonance, they will not reach to detector, and we will never be able to catch those ions. So they are very commonly used in conjunction with other gas chromatography, GCMS, or liquid chromatography MS as a simple high throughput screening system. So that's why हम अपनी mass spectroscopy के साथ ही chromatography system करते हैं, because that will help us to isolate a particular target proteins or particular family of protein depending upon their properties. So agar if we are doing something where we are aware of what we have to study, then we can couple this quadrupole mass spectroscopy with GCMS or LCMS depending upon what we want to read. So these are that's why it is called high throughputs, or that's why it's called advancement of mass spectroscopy, because we can combine them with different type of sorters, because with the help of chromatography you are actually sorting your proteins, you are selecting your pool, right? So you can use that, you can combine these techniques so that you will have your more resolved spectra, and whatever you want to study you can study that. Then quadrupole can also be placed in a tandem to enable them to perform fragmentation study. Tandem tags भी कर सकते हैं quadrupoles के साथ. The most common setup in the triple quadrupole mass spectroscopy, which enable basic ion fragmentation study, tandem mass spectroscopy, mass by mass, MS by MS, which is to be performed. So you have QQQ. अगर मैं इसको simple language में बताऊं, so what is happening here? You have three quadrupole system where you are actually selecting, consider it like a gate. I have already told you this thing. Consider it, all the three Qs are gate, some kind of a gate where you are doing screening. Screening one is removing the proteins that you do not want to study, right? Screening 2 is trying to help you select a particular kind of a protein that you want to study, and screening 3 is resolving the proteins that you want to study, like that. So you can combine them with other kind of spectroscopy, like you can do MS by MS, tandem mass spectroscopy. You can do all these things depending upon what kind of a data you want to get from your mass spectroscopy. That's why workflow of mass spectroscopy can be different from each one of you depending upon what you want to study. So you may change, you may play with the techniques, and then you standardize for your samples, right? So this is how it is. Then you have three type of, when we do triple Q, triple quadrupole based targeted proteomics, then we have these kind of selections. So first one is selected reaction monitoring, where where you are selecting a fragment ion from a single precursor and then measuring its quantification and then doing its quantification. So what is here? So for example, you have these many proteins. You have selected the green one. Now you want to know the details of this green one, right? And then you send it to a second a second quadrupole system where this particular protein is getting fragmented, ionized, further fragmented and ionized, and then from here also you are selecting few of its fragment and then studying it. So basically यहाँ selection हो रहा है. This is selection where you are selecting a particular ion for its detailed study. So selected reaction monitoring, where you are selecting, you're choosing single, you on each stage you are choosing something. Fine. In case of parallel reaction monitoring, in parallel reaction monitoring where a single precursor ion and entire fragment ions are selected. So यहाँ पर हमने क्या किया, हमने एक single precursor को select किया, उसके सारे fragments देखे और उन fragments से भी select किया. यहाँ पर हमने एक single precursor को select किया, but we are studying all its fragment. We are not choosing, we are parallelly studying all the fragments that are there. So this is a very small difference between selected reaction monitoring and parallel reaction monitoring. In selected, you are not just selecting the ion, but you are also selecting its daughter ions, its fragments. But in case of parallel reaction, you are selecting a particular ion, but you are studying the entire, all the fragments that it's going to make in the second chamber. You're studying all of them. Then you have data independent acquisition, which we have talked about in the previous this thing also, that in data independent acquisition, we divide our data into some mass to charge range. And depending upon that range, we select our ions and send it for further fragmentation to study. So that is data independent acquisition, which is DIA. Clear? Any doubts so far? So in data independent acquisition, you're not going to study single precursor, rather you're going to study multiple precursor ions, which further going to get fragmented, and you're going to read all the fragmented ion from there. So यहाँ पर आप single precursor select नहीं कर रहे, there is lot many parent precursor जिनके आप daughter ions को भी study कर रहे. Right? So this is more in detail. Data independent acquisition is dependent upon the predefined ranges that you defined before doing your experiment. Data dependent, we have also talked about data dependent. There you have, on the basis of data, you are selecting few proteins and then you're going to study them further. So this is there. Clear? Let's talk about selected reaction monitoring in detail. So this figure actually giving you the overview of SRM based targeted proteomics. So if you could see in label A, peptide mixture separated by liquid chromatography is ionized and electrospayed into the mass spectroscopy. So whenever we do any kind of a target proteomics, we always have first discovery proteomics. So we send our entire data first into discovery proteomics. There you select certain things. So here they have selected through liquid mass spectroscopy, they have selected their separated their protein mixture that they want to analyze, and then ionized it and then electrospayed it and then send it to mass spectroscopy. Specific peptide precursor is first filtered by Q1. So here if you could see, they have filtered first one. फर्स्ट Q1 में ही हमारा पहला फिल्टर लग गया है कि हमें सिर्फ इस प्रोटीन को पढ़ना है. इन फिल्टर 2, this particular in Q2, this particular protein or precursor that we want to study going to get fragmented. So Q2 में fragmentation हुआ, then specific resulting product ions are further filtered by Q3. अब इसमें से भी हमने फिर सेलेक्ट किया एक को कि हमें सिर्फ Q3 में इस पर्टिकुलर प्रिकर्सर को ही स्टडी करना है, and then once it's got selected, it is getting detected by the MS detector. So here you are selecting everything. On each stage you are selecting something. So comparing to traditional shotgun proteomic, instead of recording full mass spectrum from the protein sequence database searching, SRM only records specific precursor product transition within small mass to charge window. So if you could see, ये complete spectra है, इसमें सारे proteins की study करी है. But when you do SRM, only one precursor you have studied in detail. So this will enhance your sensitivity to a next level, and it is very specific to a particular protein. So you are getting specificity, you are also getting sensitivity. Right? So whenever you do high throughput mass spectroscopy where you want to study a particular protein or you want to do a targeted proteomics, then you utilize these approaches. Great. Next. So selective reaction monitoring, which is SRM, it is also called multiple reaction monitoring. It is a highly specific and sensitive mass spectrometry technique that can selectively quantify a compound within complex mixtures. This technique uses triple quadrupole MS that firstly target the ion corresponding to the compound of interest. तो सबसे हर स्टेज पे हम सिलेक्शन कर रहे हैं. So firstly ही it is actually targeting ions to the compound of interest with subsequent. Once you have selected your compound of interest, then you are sending it for subsequent fragmentation so that you can do a detailed analysis of the range of daughter ions you will get. Once you have range of daughter ions, in case of SRM, you will again going to select a particular daughter ion or ions. You can select 2 or 3 depending upon what you want to study. You can select one or more, but at the same time, but you are doing a kind of a selection. So you have range of daughter ions, but you are selecting some fragments from there, and then you are sending it for quantitation. Only compounds that meet both these criteria, that is specific parent ion and a specific daughter ion corresponding to the mass of the molecule of interest, are isolated within the mass spectroscopy. So you're doing, first you are selecting your parent ion, then you are from parent ion you are making a lot of daughter ions, and from daughter ions you are again making a selection. Then by ignoring all other ions that flow into the mass spectrometry, the experiment gains sensitivity whilst maintaining acquisite accuracy. There is a high level of accuracy here as well as sensitivity. Why? Because you are selecting it. You know that you want to study a particular protein only and a particular fragment only. So this is there. SRM is performed for selection of specific analyte and absolute quantification of protein, peptide, metabolite, lipids from plasma, serum, and other biological sample. So as this is, I have already told you that whenever you want to do a selected study, specific analyte you want to analyze, then you go for SRM. Then you have parallel reaction monitoring. In case of parallel reaction monitoring, you have your uh, initial proteome, you are selecting a particular ion, you are letting that particular ion undergo fragmentation, and then you are reading all the fragments that you're going to get through this particular precursor ion. So you have one parent ion but lot of daughter ion to study. In case of parallel reaction monitoring, you are studying all the daughter ions, but you have already selected your precursor or parent. So perform full scan of each transition by the precursor ion, parallel monitoring of all fragment from the precursor ion. So parallel monitoring of all daughter ions is happening in case of parallel reaction monitoring. PRM uses quadrupole Q1 to select a precursor ion. So in Q1 you are selecting it, Q2 you are doing fragmentation. The parent ion is getting fragmented, all possible daughter ions, and then in Q3 you are reading all the daughter ions. So replace the orbit trap by Q3, scan all product ion with high accuracy and resolution. Mass accuracy reached to ppm level in this case. No need to optimize fragmentation strategy because you don't want to select any fragment, you want to read all the fragments. So you don't need to have any kind of a strategy for doing fragmentation, that you may lose your sample or you may lose your fragment. That is not happening in this case. Okay. Then parallel reaction monitoring is an ion monitoring technique based on high resolution and high precision mass spectroscopy. The principle of this technique is comparable to SRM and MRM. I said that these both the techniques are quite similar. The only thing is, the only difference is you are studying only single daughter ion there, but here you are studying complete all the daughter ions. But it is more convenient in assay development for absolute quantification of protein and peptides. So SRM में हम SRM is more favorable for absolute quantification rather than relative, but it may use for both. ये मैंने आपको लास्ट क्लास में भी बताया था कि दोनों में ये यूज हो सकता है, but it is more accurate when you are doing it absolute quantification of the protein. It is more suitable for quantification of multiple protein in complex sample within with at mole level detection. So you can go to a very small amount. So all these techniques are very good for low abundant protein. So you can detect them at a very small amount, but at the same time, you do quantification of multiple protein by using such technique. PRM technology is novel technology for verification of antibody-free proteins, which is used for verification of relative quantitative proteomic data such as label-free, ITRAC, TMT, SILEC, and for targeted quantitative analysis of target protein in complex samples. So PRM के through you can verify your data, you can do the verification of data that you got data that you got from label-free iTrack, TMT, or SILEC approaches, where you are studying the entire proteome, and now you want to know what all proteins are antibody-free proteins. So in that case, you can use PRM technology. In many cases, you can do the PRM technology to verify your previous mass spectroscopy data, right? Or maybe resolve your mass spectroscopy data more. Then PRM is based on Q orbital trap as the representative quadrupole high resolution mass spectrum platform. Unlike SRM, which perform one transition at a time, PRM perform a full scan of each transition by a precursor ion, that is parallel monitoring of all the fragment that from the precursor ion. Definitely PRM gives you more opportunity to understand the changes happening in your sample, because it is reading all the daughter ion, unlike SRM एक एक टाइम पे एक ही चीज को स्टडी कर रहा है, that make it more time consuming task sometimes, right? So for P, but when you don't want to study the entire daughter ions and you want to study a particular fragment only, then SRM is the good approach for you, right? But PRM is done when you want to study lot many precursor at the same time. First, the PRM uses quadrupole Q1 to select the precursor ion, and then the selection window is usually mass to charge, which is less than 2. This is what the Q1 do. Then Q2 help in fragmentation, and Q3 help in understanding all the fragmented or daughter ions with high resolution and high accuracy. This is how it works. Therefore, PRM technology not only have a SRM, MRM target quantitative analysis capability, but also have a qualitative ability. Quality भी अच्छी है, quantitative भी अच्छी है with PRM, because you are studying all the ions that are going to get fragmented from your selected precursor. The mass accuracy can reach to ppm level, which can eliminate the background interferences and false positive better than SRM and MRM. इस केस में ज़्यादा accuracy के chances हैं, and improve the detection limit and sensitivity in complex background effectively. Full scan of the product ions without the need to select the ion pair and optimize the fragmentation energy easier to establish था इससे. Full scan करता है, हमें कोई particular हमें two selection करने की कोई ज़रूरत नहीं है. We can just select our precursor ion and get the detail, all the detail of the precursor ion, and that's how it is more easy to establish. A wider linear range increase to 5 to 6 order of magnitude. मतलब it give a wider range but a linear, as in complexity तो reduce करी है इसने, resolve कर दिया आपके स्पेक्ट्रा को, बट वाइड वे में रिजॉल्व किया, तो आप एक ही स्पेक्ट्रा से काफी कुछ पढ़ सकते हो. बट इन केस ऑफ SRM, where you are doing selection at each stage, you will definitely going to have a narrow range of spectra, because you are doing selection. Two selections are done at in case of SRM, right? So this is again, this is just a cartoon to show what is the difference between data independent acquisition and PRM. So in data acquisition, you are doing study of multiple precursor. In case of parallel reaction monitoring, you are studying a particular precursor and all the fragments that particular precursor contain. But in case of DIA, you have multiple precursor and then there are multiple daughter fragment that you're going to study. So it will be more complex data, it will be more resolved spectrum, it will be more complex system. कॉम्प्लेक्स स्पेक्ट्रा होगा इसका DIA का. बट PRM में आपको रिजॉल्व स्पेक्ट्रम मिलेगा, यू विल गेट मोर इन्फॉर्मेशन. रिजॉल्व वाइड रेंज होगी बट रिजॉल्व होगी. Then talking about targeted proteomics, so how are you going to select your peptide and how do you work? How do you choose? How do you do your assay development? This is all is given in this particular slide. So be it whatever your this thing is, whatever approach you're going to use, you first need to do a selection of signature of proteolytic peptide. So peptide should be unique to the protein of interest. Peptide should be easily detected by LC-MS analysis. Peptide length should be about 5 to 25 amino acid. both end of the peptide match enzymatic cleavage site, that is like trypsin अगर आप यूज कर रहे हो तो दोनों साइड से कट होना चाहिए. Avoid missed cleaved site, avoid wrecked, and ये नहीं होना चाहिए कि दो अलग अलग enzymatic sites हों. Then for ये this is all your experimental part, that how you going to select your this thing proteolytic, कौन सी proteolytic peptide आप यूज करोगे. सो वो सब चीजों के सिलेक्शन आपको प्रायर करने के होते हैं इन केस ऑफ टारगेटेड प्रोटियोमिक्स. अवॉइड फ्रेगमेंट frequent modification amino acid like methionine oxidation, deamidation, alkylation, phosphorylation. ये सारे जिन जिन पेप्टाइड में ये सारे मॉडिफिकेशंस होते हैं उनको आपको अवॉइड करना है, वो वाला रीजन आपको नहीं लेना है. सम केमिकली इंड्यूस्ड मॉडिफिकेशन आर आल्सो देयर. देन मिथ्योनिन ट्रिप्टोफैन के केस में ऑक्सीडेशन हो जाता है, एंड टर्मिनल ग्लूटामेट के केस में पॉलीग्लूटामिक एसिड बन जाता है इन एसिडिक कंडीशन. Likewise, you have some condition. So you have to check all these things before making your peptide selection. Then you have peptide fragment selection. You have that is specific to your peptide of interest. Select Y ions with higher mass to charge ratio. There are various contaminant ion, solvent ion, which has low mass to charge ratio. So you have to make sure कि ज़्यादा बड़ा fragment लो आप peptide का, और depending upon high mass to charge ratio ज़्यादा resolution भी मिलेगा rather than taking any low mass to charge ratio. Then B ions are often of low abundance or absent in the triple quadrupole fragment spectra. Then mass to mass signal intensity, ये भी आपको चेक करनी होती है. ये सारी चीजें ना आपको एक पर्टिकुलर सॉफ्टवेयर में डिटेल्स मिल जाती हैं. यू हैव लॉट ऑफ डेटाबेस जो आप सिलेक्ट करते हो, बट दिस इज हाउ यू चूज, दिस इज व्हाट योर experimental design is, how you going to design your mass spectroscopy, how you, but all prior knowledge you want to gain, this is what इसका ही overview दिया है मैंने ये. Optimization of each transition to maximize signal response sensitivity. So basically collision energy आपको चेक करनी है. You need to optimize collision energy for each transition. That may increase overall sensitivity, however is only really needed if sensitivity is very low or below the limit of detection. So basically you need to gather as much information of your selected peptide, so that whenever things go wrong you may check all these things. So for example, if you have you need to understand the collision energy, and especially when you are studying low abundant protein, then उस केस में तो आपको और ध्यान देनी है चीजों का. Then this is again the base collision energy equation जो कि TSQ LTS में दिया है. सर ने बहुत डिटेल्स में बताया है अपनी वीडियो में. मैं स्टिल आपको एक ओवरव्यू दे रही हूँ that you can use some template method जो कि थर्मो ने इस पर्टिकुलर सॉफ्टवेयर में दिए हुए. Likewise you can use Skyline to get your base collision equation. So depending upon that आप सेलेक्ट करते हो अपने पेप्टाइड्स को. दीज आर सम इक्वेशन जिसके अंदर आप अपनी पेप्टाइड की डिटेल्स पुट करके देखते हो, एंड देन यू सेलेक्ट योर पेप्टाइड दैट व्हाट ऑल आयंस यू वांट टू स्टडी, जो कि हम टारगेटेड प्रोटियोमिक्स में करते हैं. देन यू हैव वैलिडेशन ऑफ योर एमआरएम एसे. वैलिडेशन में क्या है, बिकॉज यू आर सेलेक्टिंग, यू आर डूइंग अ सिलेक्शन दैट यू वांट टू बी श्योर दैट वट एवर यू आर सेलेक्टिंग is that particular peptide only. So वो सब आप करते हो. वो आप कैसे करते हो, जब आप अपना पूरा स्पेक्ट्रा आपको मिलता है, because that spectra will give you details of your amino acid present in a particular peptide fragment. फिर आप उनको combine करके check करते हो that whether this is the peptide that you want to study. So these are the ways that where you do validation and you have your proper data. Then extraction of final coordinate of mRNA include selected peptide and charge state, peptide fragmentation, corresponding mass to charge ratios, fragment intensity ratio, collision energy for each transition, chromatographic illusion, time to be optimally used in scheduling SRM और MRM एनालिसिस. सो बेसिकली इसमें क्या कर रहा है कि हर चीज आप सेलेक्ट करते हो, हर चीज आप देख के रखते हो पहले से कि आपने कौन सा पेप्टाइड लिया है, उसका चार्ज स्टेट क्या है, उसके फ्रेगमेंट्स क्या क्या बनेंगे, उसका मास टू चार्ज रेशो क्या होगा उन फ्रेगमेंट्स का. इन केस उस फ्रेगमेंट का मास टू चार्ज रेशो ये है तो उनकी एक्सपेक्टेड फ्रेगमेंट इंटेंसिटी क्या होंगी, क्या मुझे उसी टाइप का स्पेक्ट्रा मिल रहा है. ये सब सारी चीजें आप एमआरएम और एसआरएम के केस में और पीआरएम के केस में ये सब चीजें आप स्टडी करके रखते हो. देन यू हैव कि कितने ट्रांजिशन नंबर होंगे, इनकी पूरी कैलकुलेशन है। इसकी डिटेल के लिए मैंने एक लिंक डाला है इस पर्टिकुलर प्रेजेंटेशन में। आप इस पर्टिकुलर साइट पे जाके ये सारी डिटेल्स डिटेल में पढ़ सकते हो, राइट। सो इस चीज के लिए आप उस पे चेक कर सकते हो। Then quickly I'm switching towards the application of PRM in proteomics research. So parallel reaction monitoring, क्योंकि हम बहुत सारा स्पेक्ट्रा एक साथ पढ़ रहे हैं। Why? Because we have selected the precursor, but we are studying all the daughter cells. Has emerged out as a powerful tool for precision and targeted proteomic analysis in proteomic research. By combining the specificity of selected reaction monitoring with the high resolution capability of mass spectra, PRM enables accurate quantification and characterization of a specific protein with complex biological samples. तो क्या हो रहा है, आप खुद सोचो, हम पहले पढ़ रहे थे अपना whole proteome. अभी तक when we were not doing any kind of a complexity, we are not getting into any kind of a complexity where we are not sorry, we are not reducing the complexity, rather we are studying the entire proteome. तब हमारे लिए बहुत difficult था to say that it is a accurate quantification or it is a highly resolved spectra, because we have lot many things there. But here you are selecting your precursor and then you are studying that precursor in detail. So accuracy थोड़ी high है, quantification correctly है, characterization of protein ज़्यादा अच्छे से होता है, and इसी वजह से PRM की diverse applications है। क्या क्या डाइवर्स एप्लीकेशन है? सबसे पहले हम कर सकते हैं क्वांटिटेटिव प्रोटियोमिक। क्वांटिटेटिव प्रोटियोमिक PRM allow for the accurate quantification of the target protein across the differential experiment condition or biological sample. So whenever we talk about quantification, we are generally accurate quantification, we are generally trying to understand what all proteins are present and how much they are present. So जब हमें ये सारी स्टडी करनी होती है तो हम PRM ले सकते हैं, क्योंकि we have selected precursors, we have selected proteins, and we are doing quantification of that protein only. By monitoring predefined set of peptide corresponding to a specific protein of interest, PRM enable researcher to investigate changes in protein expression level, post-translation modification and protein-protein interaction with high precision. So because हमारे पास already predefined set of peptides हैं, हमें वही पढ़नी है और उन्हीं के ही daughter ions को resolve करना है, PRM के थ्रू हम प्रोटीन एक्सप्रेशन लेवल स्टडी, पोस्ट ट्रांसलेशन मॉडिफिकेशन, प्रोटीन प्रोटीन इंटरेक्शन बहुत ज्यादा प्रिसाइज मैनर में स्टडी करते हैं. Then we have biomarker discovery and validation. So with the help of PRM, you can do a biomarker discovery study to identify and validate candidate biomarker from various disease and physiological condition. क्योंकि already हम complexity reduce करके we are just studying single precursor, उस precursor के जो daughter ions हैं हम उन्हीं को डिटेल में पढ़ रहे हैं. Thus it become more easy, क्योंकि हमने इतना ज्यादा रिजॉल्व कर दिया है स्पेक्ट्रा, that we can select or we can get our biomarkers here, right? So we can select that biomarkers, and that's how this particular technique helps in बायोमार्कर डिस्कवरी एंड वैलिडेशन भी बहुत ईजी है, क्योंकि हम एक पर्टिकुलर प्रिकर्सर को सेलेक्ट कर रहे हैं, तो उस प्रिकर्सर को जब भी मैं रिप्रोड्यूसिबिलिटी ऑफ दिस पर्टिकुलर सैंपल करूंगी तो वो भी एक तो ज्यादा होगी, और प्लस हम वैलिडेट कर सकते हैं हमारे और टेक्निक्स से निकले हुए बायोमार्कर्स को with the help of PRM. So for example, if I have used Silek approach initially to get the details of my sample, and now I want to study a particular protein using targeted proteomics through PRM, then it will help to validate my previous result. So PRM facilitate the identification of potential diagnostic, prognostic and therapeutic biomarker from complex biological samples such as blood, urine, tissue samples. So यहाँ से आप निकाल के आप validate कर सकते हो अपने, and you can generate a data that will validate that these proteins can be a particular diagnostic, prognostic or therapeutic biomarker. Then you have pharmacokinetics and drug target validation. So definitely with the help of PRM, you can study the changes happening in the untreated and treated. So it plays a crucial role in pharmacokinetic study and drug development by enabling the quantification of drug metabolite, protein target, pharmacodynamic biomarkers in biological matrix. So whatever your drug is targeting, so you can show with the help of PRM that जो drug treated है और जो untreated है उनमें क्या changes हैं, and that will give you more precise or rather validate your drug as a inhibitor of a particular protein. So PRM assays can be tailored to monitor drug concentration, assess drug metabolism pathway, and validate drug target with high specificity and reproducibility, providing valuable insight into drug efficacy, safety, and mechanism of action. Yeah, because हम इस PRM के केस में not only you are selecting a particular target, and if you have samples with treatment of different concentration, and when you're comparing with untreated, you may assess the drug का metabolic pathway क्या है, whether it has a good efficacy or not, reproducibility कितनी high है. I mean, you can have a higher reproducibility of your data with the help of PRM, क्योंकि already you have selected a precursor and now you are doing a detailed analysis of this particular precursor. So it becomes very easy to reproduce your data when you are having PRM approaches for your drug target validation. Then you have protein quantification in clinical samples. So PRM has significant utility in clinical proteomics for quantitative analysis of protein associated with disease state, patient stratification and treatment response. So clinical sample में तो बहुत ज़्यादा use करती है. So for example, when you're comparing normal versus cancer or normal versus disease, so particular protein को ही आप study करो, you have your targeted proteomics in the mind, so you can use PRM uh, technique in your clinical sample, because PRM ऐसे clinical sample में ज़्यादा easy to maintain होता है, because you have already selected your pool of protein that you want to study, and now you just have to apply all these the same set of protein to all your clinical samples. So it make it more uh, A, you're getting a detailed spectra, B, you can increase your reproducibility of data, right? So such a patient-derived tissues or biofluids or researcher can accurately measure protein, uh, accurately measure protein biomarkers, monitor disease progression, evaluate therapeutic intervention with clinical relevance and translational impact. So जब आप इनसे ये targeted proteomics PRM के through करते हो, you can get more detail of your protein changes that are happening, and then you can exploit these detail to have a translational research. So these all things you can do using PRM. So अगर मैं in short nutshell में बोलूँ, so verification कर सकते हो आप, validation कर सकते हो, I track differential protein का, आप label free differential protein का verification कर सकते हो. All these things can be validated using PRM. So whatever data you are getting from your basic mass spectroscopy technique, you can validate it with a better accuracy and better sensitivity using high throughput mass spectroscopy like PRM. Protein and peptide absolute quantification, quantification of disease marker, the establishment of diagnostic model, phosphorylation of protein quantification, methylation of protein quantification, all these things can be done using PRM. Other post-translational modification protein quantification can also be achieved. Quantitative analysis of pathway can be done, right? So these are the basic application of the PRM is a very crucial technique, and these are its application. It's wide. It's wide in terms of validation, verification, and it gives huge validity and reproducibility to your data. Then application of SRM in proteomics, because it's a selected one. So what all things you can do when you are doing a selected reaction monitoring. Then in case of SRM or MRM, technique enable precise quantification of a protein across the complex biological sample. तो आप एक particular protein को ही पढ़ रहे हो, और उसी particular protein को, उसी particular daughter ion को, because you are doing selection at 2 stages, आप सारे biological sample में वही सब चीजें कर रहे हो. So for example, if you have 100 clinical sample, you are just studying a particular daughter fragment of a precursor protein, and that will help you understand the disease mechanism, सेलुलर प्रोसेस और थेरेपेटिक टारगेट को भी समझने के लिए हेल्प करता है, उनकी क्वांटिफिकेशन और वो एक्सप्रेशन चेंजेस बताने में हेल्प करता है. सो दिस इज ऑल्सो इम्पॉर्टेंट, बट when you have a very specific thing to read in your proteome, then you go for SRM. When you have a particular daughter fragment in your mind. So researcher leverage SRM, MRM to quantify protein expression level, post-translation modification in protein-protein interaction with exceptional accuracy and sensitivity. क्योंकि यहाँ पे अब अगर आपको इतनी प्रायर नॉलेज है that you are even aware कि आपको कौन सा डॉटर आयन फ्रेगमेंट पढ़ना है, then you have to go for SRM and it give you equal accuracy, equal sensitivity that you get from the PRM. Because यहाँ पर अब आपका ऑब्जेक्टिव चेंज हो गया है. ऑब्जेक्टिव क्या चेंज हो गया कि आप एक पर्टिकुलर प्रिकर्सर आयन पढ़ रहे हो और उसका भी एक पर्टिकुलर फ्रेगमेंट को ही स्टडी कर रहे हो, तो फिर ना हो आपके पास और हाई चांसेस ऑफ एक्यूरेसी एंड सेंसिटिविटी होंगे. Sorry for this. So by elucidating the dynamic proteomic landscape, SRM, MRM facilitate biomarker discovery, drug development, and personalized medicine, ultimately advancing our understanding of heart disease. So these are the application. क्योंकि आप SRM के थ्रू आप एक पर्टिकुलर प्रोटीन को पढ़ रहे हो, जो प्रोटीन बायोमार्कर हो सकता है, जो प्रोटीन ड्रग टारगेट हो सकता है, जो प्रोटीन के अंदर चेंजेस हो रहे हैं, दैट्स व्हाई यू कैन टेक दैट प्रोटीन for a personalized medicine. So these techniques are actually helping you to take translational research further, a translation aspect of a particular uh, research. So you can take these proteins for biomarker discovery, drug development, and personalized medicine. Because when you are handling clinical sample, then with the help of SRM or PRM, you can have some concrete or translational data. देन मेटाबोलॉमिक्स एंड स्मॉल मॉलिक्यूल एनालिसिस भी करते हैं हम. मेटाबोलॉमिक्स स्टडीज के लिए भी SRM, MRM और PRM तीनों ही बहुत इंपॉर्टेंट है. लाइकवाइज यहाँ हम प्रोटीन के लिए कर रहे हैं, वहाँ हम मेटाबोलाइट के लिए करते हैं. तो हम इसके थ्रू मेटाबॉलिक पाथवे समझ सकते हैं, डिजीज बायोमार्कर्स वी कैन गेट, ड्रग मेटाबॉलिज्म वी कैन अंडरस्टैंड. ऑल दीज थिंग कैन बी डन यूजिंग SRM as well. Then you have a software called Skyline. It is a free software tool to build and analyze SRM, PRM, and DISA. It is freely available. Okay, you can get this particular software and you can have इस पर्टिकुलर सॉफ्टवेयर का एम ही यही है टू एम्प्लॉय कटिंग एज टेक्नोलॉजी फॉर क्रिएटिंग एंड इटरेटिंग रिफाइनिंग टारगेट मेथड्स for large scale proteome study. So with the help of this particular software, you can actually define your workflow. You can get your data from the discovery MS and you can take it further for PR. You can actually validate through PRM, SRM, or any other approach that you want to take up. So this software is very important when you talk about high throughput or proteomic studies, right? So you can use this software. The software's window look like this. So you have a Skyline, you can register it, you can download the tool for your Windows, and with the help of that you can get the details of your proteome. So you can run your pre-run raw data in this software, as well as you can retrieve the data from other databases and use this software to get the details. With the help of these details, you can decide whether you want to go for PRMs, SRM for your samples. So this software helps in all these things. Now I have already also given you, as I've mentioned in the beginning also, that this is a source where you can go and study about SRM, PRM, quadrupole, triple quadrupole system in detail. So do follow this source. You will get more detail. You will get detail about the protocol that you're going to apply for your high throughput mass spectroscopy studies. So they have given everything on this particular page. So go back and check this particular page. Now quickly coming to previous year assignments. So this is a week 8 assignment and we'll go, we have 10 questions to go with. So question number 1 is, which quantification method involve the use of heavy isotope label peptide standard for accurate quantification. कौन सा क्वांटिफिकेशन मेथड यूज को हम हैवी आइसोटोप लेबल पेप्टाइड अप्रोचेस यूज करके स्टडी कर सकते हैं? बिकॉज यहाँ पर अब इसको आपको थोड़ा सोचना पड़ेगा इन टर्म्स ऑफ कि वी आर टॉकिंग हियर वी गोइंग टू टॉक अबाउट so it's this basically is talking about high throughput only. So first thing I want to tell you this question is little you can say smartly designed, so that generally people will say it's Silec, right? Because Silec use heavy isotope, but this is not this, because it's a little tricky question in terms of quantification method. Silec will not give you a quantification of your this thing. Okay, and here we are talking about accurate quantification. So हम एक targeted study कर रहे हैं, तो उस केस में हमें selected reaction monitoring ही करनी होगी, right? बिकॉज हमें पता है हम टारगेटेड स्टडी कर रहे हैं, हमें पता है हमें कौन सा डॉटर सेल्स चाहिए, डॉटर फ्रेगमेंट चाहिए, हम सिर्फ उसकी स्टडी कर रहे हैं, एंड दिस विल गिव यू हाईली एक्यूरेट quantification of your desired protein, because now you are aware of what you have to study. So answer here going to be selected reaction monitoring. Next four question is about triple quadruple mass spectroscopy, so you have to think answer in that way only. So second question is, in multiple reaction monitoring, which is MRMS analysis using a triple quadrupole mass spectrometry, what are the two quadrupole used for? दो क्वाड्रुपोल जो हम यूज कर रहे हैं वो हम किस लिए यूज कर रहे होते हैं? सिलेक्शन ऑफ अ प्रिकर्सर आयन एंड डिटेक्शन ऑफ प्रोडक्ट आयन, ionization of the sample and detection of ion, fragmentation of the precursor ion and selection of the product ion, mass analyzer of a precursor ion and detection of the ion. MRM के केस में हम क्या कर रहे हैं? MRM के केस में क्या होता है? फर्स्ट ट्रिपल क्वाड्रुपॉल में जो टू क्वाड्रुपॉल यूज करते हैं, सेकंड क्वाड्रुपॉल यूज करते हैं, वो हम सेकंड सेट ऑफ क्वाड्रुपोल्स यूज करते हैं, वो हम किस लिए करते हैं? आई हैव शोन यू इन द डायग्राम एज वेल. Quickly, I need answer for this. Okay. Okay. Right, Aranksha. It is done for the wait. This is quadruple 2 in case of MRM. It should be C. The answer should be C here. I don't know why they are given here. I think there is some confusion. The answer going to be C. The second quadrupole system is used for, I have shown you even here, for fragmentation of a selected ion, where they have given selection of, I think there's some confusion, it's going to be C. Selection of precursor ion, detection of the product ion. That is not done by quadrupole, first or third. I'll get back to this on this in the end of the, I'll check once again whether I have marked it wrong or is it wrong, because second quadrupole system is used to do the fragmentation. फ्रेगमेंटेशन सेकंड पे होती है एंड थर्ड पे भी सिलेक्ट द प्रोडक्ट आयन. सेकंड पे भी हम सिलेक्ट कर लेते हैं कि हमें ये डॉटर आयन पढ़ना है, एंड थर्ड हेल्प इन selecting that particular ion, right? So it should be C, but anyways, let's move to the next question. Which scan mode in a triple quadruple mass spectroscopy is commonly used for targeted quantification of specific analyte? Is it a full scan mode? It is a selected ion monitoring mode. यहाँ पे हम ट्रिपल क्वाड्रुपल सिस्टम की बात कर रहे हैं. कौन सा स्कैन मोड हम यूज करते हैं कि हमारी टारगेटेड क्वांटिफिकेशन ऑफ स्पेसिफिक एनालाइट हो जाए? फुल स्कैन मोड करते हैं, सेलेक्टेड आयन मॉनिटरिंग करते हैं, प्रोडक्ट आयन स्कैन करते हैं, या प्रिकर्सर आयन स्कैन मोड करते हैं? एनालाइजर पे पूछ रहा है वो कि हम कौन सा स्कैन मोड करते हैं व्हेन वी टॉक अबाउट क्वाड्रुपल मास स्पेक्ट्रोस्कोपी. Quickly, if you can tell me the answer of this particular one. So in case of whenever we are doing triple quadrupole, it is basically done for a selected ion monitoring only because you are selecting ion here. So the SIM mode is used in triple quadrupole. This technique is made like that you are doing targeted proteomics through selection. You're selecting your precursor. Next question is, what is the primary function of the first quadrupole in triple quadrupole mass spectrometry? Ionization of the sample, mass analysis. तो यहाँ पे आपको प्राइमरी फंक्शन बताना है फर्स्ट क्वाड्रपॉल का. फर्स्ट क्वाड्रपॉल आयनाइजेशन में हेल्प करता है, मास एनालिसिस एंड सिलेक्शन ऑफ प्रिकर्सर आयन में करता है, फ्रेगमेंटेशन ऑफ आयंस में करता है, या डिटेक्शन ऑफ आयन में करता है. आई थिंक दिस वन इज similar to the previous question, but answer you have to give. सो फर्स्ट क्वाड्रपॉल में हम क्या करते हैं, हम अपना पेरेंट आयन और या फिर प्रिकर्सर आयन सिलेक्ट करते हैं. तो आंसर गोइंग टू बी बी. नेक्स्ट क्वेश्चन, क्वेश्चन नंबर 5. Q2 in triple quadrupole is also called the collision cell. जो सेकेंड स्टेप होता है जो कि हम फ्रेगमेंटेशन के लिए यूज करते हैं, विच इज used to first may you do selection, second may you make a daughter ions, or you can say fragmentation. So you have to tell which of the following occurs in Q2 for ion fragmentation. Is it a collision independent dissociation, collision induced dissociation, high energy C-trap dissociation, or high collision dissociation? In all our diagrams I have shown you this thing. Okay, Akanksha. Yeah, you are right. It is collision induced dissociation. Next question, question number 16. Which approach allows for relative quantification of protein based on the spectrum counting method? कौन सी अप्रोच है जो आपको रिलेटिव क्वांटिफिकेशन ऑफ प्रोटीन स्टडी करने के में हेल्प करती है? इज इट अ लेबल फ्री क्वांटिफिकेशन, आइसोमेरिक, isobaric tag or relative absolute quantification, which is ITRAC, TMT, tandem mass tags, or stable isotope labeling with amino acid in cell culture, silent. So कौन सी स्टडी में हेल्प करती है ये? I have to get the charger quickly. Just give me a second. I'll be back. मेरे लैपटॉप का चार्जिंग खत्म हो रहा है. So here the answer going to be label free, because we have this is basically an additional question that I have added here, because we have studied about this, that what all things, which approach you can use in a better manner to study a relative study, do a relative quantification. So here the answer going to be B. Oh, sorry, A. That is label free quantification. Just give me a second. I'm just putting a charger on my laptop. Yeah. So the answer here going to be A. That is label free quantification. So for relative quantification of protein, you have to do label free quantification. This is the previous this one question. So for MRM, multiple reaction experiment, what is the ideal cycle time that should be kept for transition list of 350 transition, so that the dwell time is 6 I have given you that, right? Yeah, here. So here you have the, so here you have the formula. So this, the transition time, they have given the dwell time should not be 6 millis, should be 6 milliseconds, and they have given you 350 transition. So you have to put this thing in this particular formula. That's why I've kept this formula, and then you can get your time, scan time. Yes, you are right, Akshita. The answer is 2.1 seconds. Right. Because you have 350 transition and you have 6 millisecond time, that will make it 2100 milliseconds. That is 2.1 seconds. Next question is, which of the following are commonly used open source software for the proteomic analysis? I've just told you that. Very simple. Yeah. Skyline as well as MaxQuant. I have given you detail of MaxQuant as well. मैक्स क्वेंट भी यूज कर सकते हैं स्काईलाइन दीज आर फ्रीली अवेलेबल सॉफ्टवेयर्स फॉर ऑल द प्रोटियोमिक एनालिसिस नेक्स्ट क्वेश्चन इज which of the following amino acid residue undergo a post translation modification is associated with the addition of small peptide called ubiquitin to the target protein? Very, I think if you have read about the post translation modification, you can easily do this question. This is the amino acid that is used for all the post translational modification, where you have with the addition of small peptide called ubiquitin. Ubiquitin भी इसी particular residue पे act करता है. Whenever your ubiquitin binds to your protein, it binds to this particular amino acid residue only. Any guesses? Akanksha? Any other person wants
No,
to?
ma'am, I don't know.
Okay, so the answer is lysine. Lysine is where you found your most of the post-translation modification that is happening, as well as whenever ubiquitin bind to a protein, it binds to a lysine residue only. Lysine is the answer. Next question is, this question you have to do, basically you have to run the blast. You have to do the blast of the FASTA sequence. It's a very simple question, nothing it's not that difficult. So basically, the goal of Sarika's research project is to comprehend the function of crucial protein A in a biology of lung cancer. The following is the protein sequence. ये protein sequence थी जो उसे अपने research पे मिली. Now she wants to discover the name of protein. So ये पूरा blast sequence उसका है जिससे हमें सॉरी आंसर ही दिखा दिया मैंने आपको बट ये सीक्वेंस बेसिकली शी गॉट दिस वाज द फास्टेस्ट सीक्वेंस she got from her research project. Now she wants to know what is the role, what is, so whenever you have your proteome, you get this, you get your data in the form of sequence, right, in the form of protein sequence. What do, you directly put the sequence. So generally you have short peptide of these sequence, you align the sequence and you try to make it a complete FASTA sequence. जितने भी पेप्टाइड फ्रेगमेंट है उन सबको अलाइन करते हैं. देयर आर सम कॉमन रीजन जिनसे अलाइन करके हम एक सीक्वेंस बनाते हैं. इट मे बी अ कंप्लीट सीक्वेंस, इट मे नॉट बी अ कंप्लीट सीक्वेंस, बट ये फास्टा सीक्वेंस को आप ब्लास्ट करते हो एंड ब्लास्ट करके आपको जो प्रोटीन मिलता है वो आप देखते हो, मतलब मैक्सिमम क्वेरी सीक्वेंस आपकी कवर हो रही होती है उस प्रोटीन. सो हेयर जब आप इसे रन करोगे तो आपको ये सीक्वेंस मिलेगा. ये नॉर्मल क्वेश्चन है, इट्स नॉट नथिंग लाइक दैट यू हैव टू get any you have to understand the explanation of this question. Basically उसने अपना प्रोटियोम एनालिसिस किया और उसे एक पर्टिकुलर प्रोटीन मिला जो कि उसे लगा कि ये बहुत क्रूशियल है in case of pathobiology of lung cancer. Now she wants to know what is this protein. उसके पास सिर्फ सीक्वेंस था. सीक्वेंस के लिए हम ब्लास्ट करेंगे, ब्लास्ट में डालेंगे ब्लास्ट टूल में फास्टर सीक्वेंस, एंड जिस भी प्रोटीन से हमारी क्वेरी सीक्वेंस सबसे ज्यादा मैच करेगी, दैट इज द प्रोटीन. सो इन केस ऑफ दिस प्रोटीन यू विल गेट सार्स को स्पाइक ग्लाइकोप्रोटीन. सो ये ग्लाइकोप्रोटीन आएगा और ये बहुत इंपॉर्टेंट उसके रिसर्च प्रोजेक्ट में था. आई एम नॉट आस्किंग यू टू गो एंड डू द ब्लास्ट, यू कैन डू इट लेटर, but just giving you a glimpse of what kind of questions you have for your exam and for this week assignment. Next question is, Sagrika wants to know the uniprot ID and gene ID of the protein A. तो उसको एक प्रोटीन मिला, अब उसको अगर ये जानना है कि इसका जीन आईडी क्या है, यूनिप्रोट आईडी क्या है, what she has to do? Now you have to tell me what is the gene ID, but I'm not asking you to tell me the gene ID and the gene uniprot ID and gene ID here. I'm just asking कि अगर आपको किसी भी प्रोटीन का यूनिप्रोट आईडी और जीन आईडी जानना है तो आपको क्या करना होगा, कौन सी साइट पे जाओगे? रादर दे हैव गिवन यू द हिंट एज वेल. So what you can do is you can paste your sequence to blast. You got the name of the protein. Now you can put this the same. You can search the protein in the uniprot. When you're going to search the protein in the uniprot, you will get the uniprot ID as well as the gene ID. These are the things that you do when you analyze your proteome data. You get the sequences, you do it, you put it in the blast, you get the gene ID, you get the uniprot ID. Answer here going to be D, but the main objective of this question is to let you know that what all software you have to use while analyzing your proteomics data, right? So you get your data in the form of sequences, एंड देन उनके नंबर्स भी होते हैं, उनकी क्वांटिफिकेशन एक अलग टेबल दिया होता है, बट मोस्टली इफ यू वांट टू आइडेंटिफाई अ प्रोटीन देन द प्रोटीन आइडेंटिफिकेशन इज डन थ्रू सीक्वेंस ओनली. So you just normally run your blast or your protein blast and you get the detail of your protein. And if you want to know the ID or uniprot ID or gene ID, then you have to use some other software to get the exact ID. Okay, so this is all for the week 8. Now I just want to tell you that if you all are planning to appear for your exam, I have given you answer for each week question from the previous year. You just go through all those PPTs or videos and you will get the idea, because uh, before question answer I always give you a brief of concept, and whatever question you have in your, wait a minute Akanksha, whatever question you have in your assignment, you will get the concept of that particular question in my brief summary. Yeah, go ahead. You have any question?
Ma'am, in the week 8 assignment, there was a similar question in which we were given the protein sequence,
Hmm.
and we were supposed to identify the protein using blast. So ma'am, in the following question, in the next question, we have to identify its accession number, protein's accession number.
For the,
So
okay.
for the protein, we identified in the last question,
Okay.
we are supposed to identify its accession number.
Okay.
So ma'am, when we do blast, when we get a list of proteins
Hmm.
which
Okay.
might be there, so ma'am, we get in the last row we get accession number, and when we do the blast, and when we use uniport, we get the uniport id. So what's the difference? Like, they are asking the accession number, and in this week's question, uh, they have given this link to uniport to
Yeah,
identify the
so
uniport
basically
id.
what happened when you do so why there is so you want to know why do we want to get why there is a need to get UniProt ID, right? जो मैं आपके क्वेश्चन से समझ पाई
मैम,
आप यही कहना चाह रही हो,
हाँ
हमें यूनिप्रोट
मैम,
करना ही क्यों
जबकि
है जब हमें
नहीं
एसेशन
मैम,
नंबर
जब
है? ओके।
हाँ मैम, जब मैम उस क्वेश्चन में ही दिया है कि हमें एसेशन नंबर फाइंड आउट करना है, लेकिन फिर जो ऑप्शंस दिए हैं वो यूनिप्रोट आईडी के हिसाब से दिए हुए हैं।
वो कोई भी कंफ्यूजन हो सकता है कि उन्होंने एज इट इज प्रीवियस का कुछ ऐड कर दिया हो, ये हो सकता है,
हाँ,
क्योंकि एसेशन नंबर तो आपको नॉर्मल ब्लास्ट करने से भी
इस
मिल
पे
जाएगा।
मिल जाता है,
हाँ
यस मैम।
राइट, यस यस। सो वो हो सकता है कि वो गलती से उनसे कॉपी हो गया हो वहां पर, और दैट्स वाई वो हुआ हो। ऐसा हो सकता है। जब असाइनमेंट बना रहे होते हैं तो समटाइम यू यूज द प्रीवियस टेम्पलेट। तो जनरली आपको इस तरीके से इसको ब्लास्ट करके, मैं आपकी स्क्रीन पे ब्लास्ट ही कर रही हूँ सीक्वेंस को। सो यस,
हाँ
डेफिनेटली यू गेट द एसेशन नंबर फ्रॉम समथिंग इज रॉन्ग। So you definitely going to get your accession number there only.
मैम, ब्लास्ट पी।
ओ मैंने पी नहीं किया, सॉरी, आई एम
यस
सो
मैम।
सॉरी। हाँ राइट। सो you get your accession number there only, so you don't have to go to UniProt. वो गलती से वो प्रीवियस असाइनमेंट सेलेक्ट हुई होगी और उस वजह से चला होगा। uh, ओ मैंने ब्लास्ट कर
मैम,
दिया।
लेकिन उसमें यूनिप्रोट आईडी का ही ऑप्शन दिया हुआ है, उसमें एसेशन नंबर के
सो वो
ऑप्शंस ही नहीं
गलती
है।
से उनसे हुआ होगा, दे माइट हैव रिटन अ सेशन नंबर बट दे हैव नॉट रिटन द चेंजेस। बट सेशन नंबर के केस में यू नीड टू गिव a session number only. In case of
ओके
UniProt
मैम।
ID, you use UniProt ID वाला sequence only. So
ओके
that is
मैम।
there, but don't worry, वो कभी कभी हो जाता है, वो कोई मैन एरर भी हो सकती है, मतलब वो
यस मैम।
ह्यूमन एरर भी हो सकती है वहां पर, बिकॉज वो हुआ हो सकता है। बट अगर आपसे सेशन नंबर मांगा है तो डोंट वरी, यू विल गेट दी, you have to submit the session number only there. But अगर options में ही session number नहीं है, UniProt ID
हाँ,
है,
यस
then
मैम,
go
यस
for
मैम।
UniProt ID, right?
ओके मैम।
That's
ओके
why they
मैम।
have given you hint. वो होता है कभी कभी किसी से गलती हो गई होगी वहां पर, वो थोड़ी एरर हो जाती है इन चीजों में, right?
ओके मैम।
So if you are getting something like this, you have to get the UniProt, you have to write the UniProt ID only in your
ओके मैम।
assignment. This is for your assignment. And for your exams only, exam, you may not get such kind of a question. I don't know what all facility they're going to provide you during exam. If it's with internet, then definitely you may have to do such questions. But if it's not, if it's a written with objective questions, then you will get similar sort of question, but not this one, where you're going to use your softwares. मुझे exactly नहीं पता what kind of a pattern you have for your coming exam, but mostly questions going to be through uh, जो आपके असाइनमेंट्स में क्वेश्चंस है ना, उन्हीं से रिलेटेड क्वेश्चंस होंगे आपके, इफ यू आर गिविंग द एग्जाम। सो go through all the assignments, see all the topics that you have there, अपने सारे क्वेश्चंस का कुछ बेसिक आपको राइटअप पता होना चाहिए, आपको पता होना चाहिए बेसिक कंसेप्ट ऑफ अ टेक्निक, बिकॉज मोस्टली हमने टेक्निक पढ़ी है, मोस्टली हम आपको सारी चीजें पता, ये सब चीजें पता होनी चाहिए कि जब हम कोई पर्टिकुलर प्रोटोकॉल फॉलो कर रहे हैं, व्हाट ऑल थिंग्स वी आर यूजिंग, लाइक 2D जेल के केस में हमने कुछ चीजें यूज करी थी, व्हेन वी केम फॉर मास स्पेक्ट्रोस्कोपी एनालिसिस हमने एंजाइम्स यूज करे थे, हमने हाउ वी एक्चुअली प्रिपेयर आवर सैंपल फॉर ऑल दीज टेक्निक। ये सब चीजें आपको थोड़ी डिटेल्स में पूछी जा सकती हैं। दे हैव लॉट ऑफ there can be lot of question from this particular section where they may ask, because यहाँ पे आपकी labs भी हुई हैं online, right, where they have told you how to prepare your sample, what kind of treatment you have to give to your sample to make it ready for your mass spectroscopy analysis. So ये सारी चीजें थोड़ा सा आपको देखनी होंगी, आप प्लीज अपने सारे असाइनमेंट्स को चेक कीजिए, वो बहुत इंपॉर्टेंट है। And in case you have any other question, just ask me, and go through all your PPTs. You have the access, go through all these PPTs, try to remember the concept, and all the best for your exam. I'm here till 8. If you have any question, any query, just let me know.
Ma'am, I have a few questions from this year's assignment.
Okay.
this year's previous assignments.
Hmm.
Just a minute, ma'am.
Go ahead.
Ma'am, week one's ninth question.
Week one. Okay.
Yes,
Okay.
ma'am.
Give me a
Ninth
second.
question.
Let me go to the week one. Okay. Yes.
Ma'am, why can't be the option number second correct? Protein
Can you
folding
read the
is.
question for me
Ma'am,
because I may have different question.
ma'am, the question is which of the following statements best describes the relationship between protein folding and thermodynamics?
Okay, I may not have that question with me right now. Okay, I'm assuming. Okay, give me the options. This is this
Ma'am,
year's assignment.
yes, ma'am. This year's assignments, week one,
Okay,
this year.
then give me one more minute so that I can go to the page and get the assignment for you. Okay. Um, I may not have the just a second, one more minute. Trying to get on the this year's assignment page. Okay, I'm unable to log in. But anyways, can you share your screen or something? Maybe I'll see from there. Is it
Yes, ma'am. I
possible?
can. I can.
Yeah,
Yes, ma'am.
just share your screen so that I can see the question. I'll stop sharing here. Yeah, okay. Protein folding is an iso. Which of the following statement best describe the relation between the protein folding and thermodynamics? Okay. So protein folding is an isothermal process where no heat is exchanged with the surrounding. Protein folding is an Endergonic process require input of energy. Protein folding is driven by a decrease in entropy and increase in enthalpy. Protein folding is governed by a principle of increasing disorder. Why do you think that it should be second?
Ma'am, because initially protein folding does require input of energy.
Do you know? I don't know whether I have taken this particular concept with you or not, but your proteins are folded and remain in that state because there is no randomness and there is a stability in that protein. So some things require energy, some bond formation require energy, but entire protein folding require endo is an endogonic process and require input of energy is wrong. When we say that this particular folding happen as a spontaneous reaction, right? When you talk about spontaneity or when you talk about this thing happen naturally, it may require input of energy to make some bond, but the entire folding is not because of input of energy. क्योंकि अगर हर बार इनपुट ऑफ एनर्जी से फोल्डिंग होगी तो डू यू थिंक सेल विल एबल टू वर्क? एक बार इसको सोच के देखो, जस्ट थिंक अबाउट इट इन एन ओपन मैनर कि क्या होता है वो प्रोटीन फोल्डिंग, हमारे सेल्स कैसे काम करते हैं. कुछ चीजों के लिए you require energy, but is it for protein folding? Are you there?
Yes, ma'am. Ma'am, I'm still not able to understand.
So okay, fine. जो protein, let me give you in a little textual manner. जो प्रोटीन फोल्डिंग का प्रोसेस है वो फ्री एनर्जी यू नो.
Yes, ma'am.
सो ये जो पूरा फनल इन्होंने बनाया है ना टू शो दिस थिंग ओनली कि
Hmm.
व्हाट दे हैव शोन दैट इफ is particular I don't know if you can see I'm not sharing the screen so I can't even. तो अगर आप राइट हैंड साइड को देखोगे, राइट हैंड साइड को देखो तो देयर इज एन ओपन माउथ ऑफ फनल. ऊपर से देख रहे हो आप एंट्रोपी व्हेन देयर इज अ होल ऑफ रैंडमनेस,
Yes, ma'am.
राइट? एंट्रोपी हाई मतलब रैंडमनेस, राइट? सो फनल जो है वो आपको रिप्रेजेंट कर रहा है कि this is basically wide range of structures which are accessible to the ensemble of denatured protein. यहाँ पर जितना ज्यादा हाई एंट्रोपी होगी उतने ही आपको डीनेचर्ड स्ट्रक्चर मिलेंगे प्रोटीन के, और जितना ज्यादा आप नीचे आते जाओ जितनी रैंडमनेस कम होती जाएगी यू विल फाइंड स्टेबल स्ट्रक्चर्स. क्या ये आपको थोड़ा
Hmm.
कन्विंसिंग लग रहा है?
Yes, ma'am.
तो
Yes,
जो
ma'am.
मैंने आई थिंक आपको ये बात बताई होगी कोलैप्स स्टेट ऑफ प्रोटीन के बारे में. The initial collapse state of protein जिसके अंदर लिटिल थर्मोडायनेमिक स्टेबिलिटी होती है उसे हम मोल्टन ग्लोब्यूल कहते हैं. इस पे बेसिकली फर्स्ट टाइम जहां पर मोल्टन ग्लोब्यूल लिखा हुआ है ना, दैट इज द फर्स्ट स्टेज जहां पे थोड़ी सी थर्मोडायनेमिक स्टेबिलिटी आती है. दीज आर जस्ट some models or you can say some theories behind how your protein get uh, folded. तो हमने मॉल्टन ग्लोब्यूल स्ट्रक्चर पढ़ा था जिसमें मैंने ये बताया था कि फर्स्ट थर्मोडायनेमिक स्टेबिलिटी जो आती है किसी भी प्रोटीन की वो उस पर्टिकुलर स्टेज पे आती है व्हेन यू हैव योर मॉल्टन ग्लोब्यूल स्टेज. ठीक है, तो उसको हम फर्स्ट स्टेज कहते हैं, वहां पे थोड़ी सी स्टेबिलिटी आती है, बट एज यू गो डाउन इन दिस फनल यू हैव योर मोस्ट स्टेबल स्टेट. तो बेसिकली क्या हो रहा है, थर्मोडायनेमिक एस्पेक्ट से सोचना आपको यहाँ पे, कि जब एंट्रोपी हाई थी, व्हेन देयर इज अ ह्यूज रेंडमनेस, देयर वर लॉट ऑफ प्रोटीन्स दैट आर बीइंग फॉर्म्ड, दे ऑल आर डीनेचर्ड, they all are not stable, they are getting they are not stable in terms of वो structures बन रहे थे और टूट रहे थे. But as you go down in the funnel, where your entropy is decreasing and enthalpy is increasing, you have more stable or folded structure. समझ आया ये बात आपको?
Yes, ma'am.
राइट. सो ये है, ये एक मॉडल है बेसिकली, ये एक मॉडल रिप्रेजेंट हुआ हुआ है. एंड इस
Mm.
केस में आप इस मॉडल को देख के आप ये नहीं कह सकते कि विद द हेल्प ऑफ एनर्जी यू आर गेटिंग अ प्रोटीन फोल्डेड. यहाँ पे फ्री एनर्जी के टर्म्स में बात की है, एंड दे आर टॉकिंग अबाउट एंट्रोपी एंड एंथैल्पी. जितनी ज्यादा रैंडमनेस होगी उतना अनस्टेबल स्ट्रक्चर होगा, जितनी ज्यादा स्टेबिलिटी है मतलब रैंडमनेस सबसे कम है, and that's how you are coming to your final stable structure.
Okay, ma'am.
राइट. सो ये
Yes,
उस
ma'am.
चीज को रिप्रेजेंट कर रहा है. सो ऑलवेज लुक एट द क्वेश्चन, कि क्वेश्चन क्या बात कर रहा है. क्वेश्चन अभी इनपुट ऑफ एनर्जी की बात ही नहीं कर रहा, बिकॉज फ्री यहाँ पे कहीं पे भी ये नहीं बताया हुआ कि एनर्जी गेन से यू आर गेटिंग अ फोल्डेड स्ट्रक्चर. देर आर,
Mm.
मैंने बताया ना, बहुत सारे बॉन्ड्स है जिनको बनने के लिए एनर्जी की रिक्वायरमेंट है. बट
Yes, ma'am.
in this particular figure they are just talking about free energy, entropy and enthalpy, and you have to give answer in that way only. ऐसा.
Okay, ma'am.
Okay. Next question.
Oh, ma'am, just a minute.
Let me know the week. कौन से week का है, ताकि मैं I'll open the.
Ma'am, this is week 2, question number 5.
Okay. It is week 2. Okay, this is this year's question. Just a sec. Okay, I can see it on your screen, by the way. So we plan to conduct SDS page on 4 samples AD and begin to quantify their protein concentration using Bradford. She dissolved each protein in 50 microliter of the buffer and used 2 microliter of each sample in duplicate for quantification. So basically she has diluted the sample. 50 microliter buffer में उसने each protein sample in 50 microliter of buffer and use 2 microliter of the sample and duplicate. But कितना protein लिया? Anyways, leave it. To create a standard curve, she used BSA 2 microgram, 4 microgram. उसने standard curve बनाया. Standard plot हो गया. Now she has these values. Okay. So the absorbance of the A and D were this one. If Surbi wishes to load 15 μg protein for each sample, what would be the volume required for sample A and D? A का है इतना 0.505. ठीक है, 0.505. अगर मैं ओडी देखूं, मैंने ये क्वेश्चन शायद सॉल्व भी करवाया आपको. अगर आप ओडी देखो, दिस इज बीएसए और ये आ रही है अप्रोक्सीमेटली 8 के पास, राइट?
Yes, ma'am. Approx 8 के पास आ रहा है.
8 के पास आ रहा है ये.
Yes,
और
ma'am.
8 के पास आ रहा है मतलब ये इतना, इसके अंदर 8 म्यूजी प्रोटीन तो होगा आफ्टर दिस मच डाइल्यूशन,
Yes, ma'am.
राइट?
हाँ.
एंड लाइकवाइज 10 के लिए आ रहा है, सॉरी डी के लिए आ रहा है 2.212. मैं 4 के अप्रोक्सीमेटली
4
सोच
के
सकती हूँ.
approx. Yes, ma'am.
या
Yes, ma'am.
राइट.
हाँ.
अब अगर हम अब उसको अगर 15, सो फॉर अब इसको इस तरीके से देखो कि ए में आपकी जो ए प्रोटीन है उसमें आपकी प्रोटीन ज्यादा है या कम है, क्या बताओगे आप आपके हिसाब से? ए और डी का कंपेयर करें तो ए में
मैम,
प्रोटीन
ए में
ज्यादा है,
ज़्यादा है.
ए
ए
में
में ज़्यादा
प्रोटीन
है.
ज्यादा है और
Yes,
डी में
ma'am.
प्रोटीन कम है.
समय।
राइट? सो
मैम।
दैट डेफिनेटली डी का आपको ज्यादा लोड करना होगा और ए का कम लोड करना होगा। तो ये तो पक्का है कि ए की वैल्यू कम आएगी और डी की वैल्यू ज्यादा आएगी, राइट?
यस मैम।
ये ठीक है। नाउ
हाँ।
अब अगर आप देखो 15 म्यूजी और अगर ये अप्रोक्सीमेटली 8 म्यूजी के अराउंड है, क्योंकि अभी ये डाइल्यूटेड, अभी मैं इसको डायरेक्ट रीडिंग्स ले रही हूँ, आई एम नॉट इवन टॉकिंग अबाउट द डाइल्यूशन। अगर हमारा 15 म्यूजी हमें लेना है तो आपको ये कैलकुलेट करना पड़ेगा, ये जो वैल्यू है ना 5.505, इसको आप
यस
इस
मैम।
पर्टिकुलर ऊपर जो हमारी y इज इक्वल्स
इक्वेशन
टू
दिया है।
इक्वेशन में आप
यस मैम।
डालोगे। तो
ठीक है।
आपका इज y इज योर ओडी,
यस मैम, एक्स
एक्स
कैलकुलेट करना
कैलकुलेट करना है।
है। यस
तो
मैम।
होगा आपका 0.505 को आप डिवाइड माइनस करोगे पहले इससे एंड उसके बाद डिवाइड कर दोगे 0.064 से, राइट? एंड
यस
यू
मैम।
गेट दी एक्स। जो आपकी एक्स वैल्यू आएगी वो आपकी ए की कंसंट्रेशन होगी,
हाँ।
ठीक है? तो जो भी
यस
वो
मैम।
वैल्यू आएगी वो उसकी कंसंट्रेशन होगी। अब फॉर एग्जांपल मान लो वो आती है आपकी 7 पॉइंट समथिंग,
यस
राइट?
मैम।
तो आप
7.7।
15 म्यूजी आपको फाइनल लेना है। 15 को आप डिवाइड कर दोगे 7 से तो समवेयर अराउंड 2 के अराउंड आना चाहिए या 3 के अराउंड डिपेंडिंग अपॉन। तो आप इसमें इस ऑप्शन में देख पा रहे हो ना कि या तो 2 के आसपास वैल्यू होगी, मैंने अभी कैलकुलेट नहीं किया है, कैलकुलेट आप करना। मैं जो आपको समझा रही हूँ वो ये है कि
यस
आपकी
मैम।
जो वैल्यू आएगी दैट इज योर एक्चुअल प्रोटीन का कंसंट्रेशन।
डिवाइडेड बाय द कंसंट्रेशन जो कि हमें ओडी के पैरेलल मिल रहा है।
हाँ, मतलब जो भी आपको रीडिंग आई वो आपको एक्चुअल प्रोटीन ए की वैल्यू जो एक्स एक्स आएगा ना ए के लिए,
हम्म।
जब आप
हम्म।
वाई डालोगे ए के लिए विच इज 0.505 एंड देन यू गोइंग टू कैलकुलेट यू गेट दी एक्स, दैट इज योर ए का वैल्यू। अब ए का वैल्यू आएगा, अगर आप अभी कैलकुलेट करके देख लो, मैं एक बार जल्दी से कैलकुलेट
ठीक
करती
है
हूँ।
मैम, एक मिनट।
तो हम देखते हैं, तो ये आ रहा है 0.505 माइनस 0.0127 equal divided by 0.064. This is coming 7.69, right? Up 15
यस मैम,
divided
सेवन
by
पॉइंट सिक्स नाइन हाँ मैम, आ गया।
7.69, right?
यस मैम।
7.69, it should come 1.95, right? 1
15
point
डिवाइडेड बाय
15 डिवाइडेड बाय 7.69 करोगे
7.7।
1.59 आएगा।
ओके।
अगर मैं इसको 2 से मल्टीप्लाई करती हूँ,
हाँ।
2 से मल्टीप्लाई इसलिए किया है, शायद इसमें कुछ डाइल्यूशन फैक्टर का होगा जो कि इन्होंने बहुत करेक्टली नहीं लिखा है कि कैसे इन्होंने, जैसे इन्होंने लिखा हुआ है 50 को 2 से 50 माइक्रोलीटर लिया और उसका 2
हम्म।
माइक्रोलीटर उठाया, राइट? सो
हाँ।
बेसिकली 2 माइक्रोलीटर में कितनी होगी कंसंट्रेशन, वो कुछ है इसमें डाइल्यूशन फैक्टर। तो आप 1.9 को इंटू 2 करोगे तो 3.8 आएगा, दैट इज योर आंसर फॉर वन। अभी
ओके।
के लिए 1.9 रखते हैं, अभी के लिए 1.9 रखते हैं, आप डी को सॉल्व करो एक बार।
एक मिनट मैम।
एक्चुअली बहुत क्लैरिटी से नहीं लिखा है इन्होंने कि इन्होंने बीएसए को सॉल्व डिसॉल्व किया या प्रोटीन को, ऑल्दो प्रोटीन बताया हुआ है बट आइडियली डाइल्यूशन फैक्टर डिवाइड नहीं होता, मल्टीप्लाई होता है, बट आई डोंट नो हाउ दे हैव डन इट। बट अगर आप डी को भी सॉल्व करोगे ना तो अप्रोक्सीमेटली 4 के अराउंड ही आना चाहिए। 0.2125 माइनस 0.0127 डिवाइडेड बाय 0.064, तो ये 3.12 आया। इसको भी 3.12, आप 15 से डिवाइड करो 3.12 को।
हाँ मैम, 9.37 के
हाँ, तो
अराउंड आ रहा है।
इंटू 2 करोगे तो 9.7 आ
हाँ।
जाएगा। सम
9।
टाइम सम टाइम मैं तुम्हें ये एडवाइस दे रही हूँ फॉर सॉल्विंग एनी क्वेश्चन। कभी
यस मैम।
तुम पहले ये हमेशा क्वेश्चंस को सॉल्व करते हुए, बिकॉज कुछ चीजें ना मिसिंग हो सकती हैं पेपर में बट यू वांट टू क्लियर इट, सो
यस मैम।
क्वेश्चन को पहले अपने आंसर निकाल लो जो तुम्हारे हिसाब से आंसर आएगा, राइट? ये तुम्हें, जैसे मैंने तुम्हें अप्रोच बताया ना कि ए की वैल्यू लेस होगी और डी की वैल्यू ज्यादा होगी फॉर
हाँ
श्योर,
हाँ
तो
हाँ।
आपके जो फर्स्ट ऑप्शन है वो तो हट ही गया,
वो तो हट गया, यस
राइट?
मैम।
अब
हाँ।
और आपका नीचे वाला लास्ट वाला ऑप्शन भी हट
हट
गया,
गया।
राइट? ये दो ऑप्शन हट गए, आपके दो ऑप्शंस बचे।
दो
अब
ऑप्शंस
आपकी
बचे हैं।
वैल्यू आई 1
हाँ।
पॉइंट जो भी वैल्यू आई और प्लस जो भी वैल्यू आई, दूसरी डी की भी जो वैल्यू आई आपकी 4 पॉइंट समथिंग, राइट। 4.8 डी की वैल्यू आई, 1.9 डी ए की वैल्यू आई थी, इनटू टू।
9, हाँ
कोई
हाँ
भी डाइल्यूशन, क्योंकि यहाँ पे 2 का डाइल्यूशन किया है उन्होंने, उन्होंने 2
हाँ हाँ
माइक्रोलीटर
हाँ
सैंपल इनटू 2
हाँ।
करके देखो, आपका आंसर अगर आ रहा है तो वो आंसर आपका है।
ठीक है मैम।
राइट, इस तरीके से
हाँ।
आप ना सॉल्व करते हो अपने क्वेश्चन, जब आपको सिर्फ क्वेश्चन सॉल्व करना है। बट
हम्म।
डेफिनेटली जब आपको रिसर्च करनी है, तब आपको
हम्म।
बहुत एक्यूरेट होना पड़ेगा। और उस एक्यूरेशन में आपको ये सारे क्वेश्चन सॉल्व नहीं करने, आपको एक्चुअल में आपके रिजल्ट्स होंगे और उससे आपको रिजल्ट आएगा। आपको पता होगा आपने क्या डायल्यूशन फैक्टर यूज किया है, राइट। बट ये
ठीक है मैम।
सॉल्व इस तरीके से ही होना है। तो ये
ठीक है।
चीजें आपको ध्यान रखनी है जब आपको अपना क्वेश्चन सॉल्व करना है। नेक्स्ट
ठीक है
क्वेश्चन।
मैम। मैम क्वेश्चन नंबर 8।
You have a protein solution with concentration 2 mg per ml. You need to precipitate the protein using 10 trichloroacetic acid. If you have 1 ml of the protein solution, how much 100% TCA do you need to add to achieve the desired concentration of 10%? Okay. So अभी आपके पास, अब उन्होंने क्या है, आपके पास concentrated solution है 2 mg per ml का। ठीक है, आप यू नीड टू प्रेसिपिटेट दिस। इफ यू हैव 1 ml ऑफ द प्रोटीन सोल्यूशन, आपके 1 ml प्रोटीन सोल्यूशन है, कितना टीसीए डालोगे कि फाइनल कंसेंट्रेशन 10% आ जाए? 100% का कितना डालोगे 1 ml में? तो आपको चाहिए 10%, आपके पास है 100%, तो कितना डालोगे 10 की 10% हो जाए? एम वन एम वही करना है, M1V1।
M1V1 इक्वल्स
हाँ
M2V2। मैम आप वैल्यूज बता सकते हो पुट करके?
हाँ, तो मैं बताती हूँ। देखो आप अभी आपके पास you have 1 ml of protein, इसमें तो basically 10 going to be your M2, 100 going to be your M1, what you have and what you want basically। एक मिनट रुको, मैं इसको सॉल्व करके आपको देती हूँ। 10 डिवाइडेड बाय 100 इंटू 1 ml, ये आपका आंसर होगा। दैट गोइंग टू बी 0.1 ml। आपको समझ आया? आपको डिजायर्ड क्या है? आपकी M2 गोइंग टू बी 10% एंड V2 गोइंग टू बी 1 ml। बट आपके पास टीसीए है कितना? 100%। 100% की कितनी वॉल्यूम आपको डालनी है कि आपको 10% मिले 1 ml में? तो आपके पास V1 की वैल्यू नहीं है। अब सॉल्व करके देखो इसको।
मैम V2 की क्या वैल्यू है?
V2 1 ml।
ओके।
हो गया ये सॉल्व?
यस मैम हो गया।
0.1 आ गया इसका आंसर।
यस मैम आ गया।
ओके, एनी अदर क्वेश्चन?
यस मैम जस्ट अ मिनट। मैम क्वेश्चन नंबर 10th।
Okay, so when we do post translation modification, what are the post translation modification we study? और यहाँ पर
मैम
ये याद रखना
असाइलेशन,
कि हम,
फॉस्फोराइलेशन।
हाँ राइट, फॉस्फोराइलेशंस। और हम कर
असाइलेशन।
रहे हैं असाइलेशन, ठीक है। हम कर रहे हैं 2 डायमेंशनल इलेक्ट्रोफोरेसिस।
यस
तो दे
मैम।
आर आस्किंग कि कौन सी टेक्निक को हम इंटीग्रेट करेंगे 2 डायमेंशनल इलेक्ट्रोफोरेसिस के साथ टू स्टडी
हम्म।
पीटीएम। इट इज बेसिकली फॉस्फोराइलेशन इन फॉस्फोप्रोटीन विच इज फॉस्फोराइलेशन प्रोटीन इनरिचमेंट ओनली। फॉस्फोप्रोटीन का मतलब है, या फॉस्फोप्रोटियोम का मतलब है फॉस्फोराइलेशन प्रोटीन विथ फॉस्फोराइलेशंस। राइट, सो बहुत क्लियर आंसर है ये। ग्लाइकोप्रोटीन स्टेनिंग क्यों करेंगे हम?
मैम मैम ग्लाइको ग्रुप भी तो ऐड होता है ना जब
ग्लाइको
हम
ग्रुप
लोग।
ऐड होता है हम यहाँ पे टू डायमेंशनल इलेक्ट्रोफोरेसिस कर रहे हैं।
यस मैम।
टू डायमेंशनल इलेक्ट्रोफोरेसिस में what all things you can study for when we talk about post translation modification? ग्लाइकोप्रोटीन जो आप कह रहे हो ना कि ग्लाइकोप्रोटीन स्टेनिंग कर सकते हैं हम,
यस
this
मैम।
will not give you post translation modification come in a reaction form. Glycosylation, glyco proteins add होते हैं, but at the same time in response to specific treatment बोला मैंने, तो when we talk about any treatment, phosphoprotein enrichment is the only, I think these are the things that we study with 2DE. क्योंकि रेस्ट जो है हमारा, वो हम टू डायमेंशनल से स्टडी ही नहीं कर पाते। मेरे को दिस इज माय ओपिनियन, आई नीड टू चेक दिस थिंग, जस्ट गिव मी अ सेकेंड। आई थिंक it should be phosphoprotein enrichment only, but glycoprotein glycoprotein is staining is not, just a minute, ग्लाइकोप्रोटीन स्टेनिंग से वी विल नॉट गेट दिस थिंग, पोस्ट ट्रांसलेशन नहीं मिलेगा। ग्लाइकोप्रोटीन स्टेनिंग इज डन फॉर ग्लाइकोसाइलेटेड प्रोटीन बताता है ये, बट अगर हमें इन्वेस्टिगेट करना है, देखो ग्लाइकोसाइलेशन is a part of structural changes that happen in the protein after translation, right?
यस मैम।
So it, अगर मेरे पास दो ऑप्शन होते तो मैं शायद दोनों ही लगाती, बट मुझे ऐसा लगता है फॉर टू डायमेंशनल वी डू फॉस्फो, फर्स्ट थिंग फॉर पोस्ट ट्रांसलेशन मॉडिफिकेशन what we do in two dimensional electrophoresis is phosphoprotein enrichment, because we want to check proteins that are undergoing phosphorylation.
ओके मैम।
ठीक है, तो ये थोड़ा सा, आई थिंक मैंने बहुत कन्विंसिंग आंसर नहीं दिया होगा तुम्हें, बट ये जो ग्लाइकोसाइलेशन है, दीज आर ऑल स्ट्रक्चरल चेंजेस।
ओके मैम।
सो आई डोंट नो इसको मैं किस तरीके से बोलूं, बट whenever we do talk about 2DE, it has to, for post translation modification, it has to be phosphoprotein enrichment. That is what we can do. I don't know how we do glycoprotein staining when it comes to 2DE. मुझे इस चीज़ की थोड़ी नॉलेज नहीं है, क्योंकि टू डायमेंशनल के केस में वी आर सेपरेटिंग प्रोटीन ऑन द बेसिस ऑफ आइसोइलेक्ट्रिक फोकसिंग। उस केस में वी कैन ओनली डू वी कैन ओनली एसोसिएट दिस टेक्निक विथ फॉस्फोप्रोटीन एनरिचमेंट। अदरवाइज और चीज़ें मुझे पता नहीं ग्लाइकोप्रोटीन कर भी सकते हैं 2D के साथ कि नहीं। आई हैव माई डाउट्स, बिकॉज मेरे को लगता है वो सिर्फ वन डायमेंशनल में चेक करते हैं, बिकॉज 2D में सेपरेशन कर तो उसमें भी सकते हैं, क्योंकि एनरिचमेंट, ओ सॉरी रेजोल्यूशन तो उसमें भी हो रहा है, बट
यस मैम।
आई हैव माई डाउट कि वो नेटिव कंडीशंस में करते हैं या रिड्यूसिंग जेल में करते हैं। आई हैव माई डाउट्स ऑन दैट। सो when it comes to post translation study, just first go with the phosphorylation studies.
ओके
क्योंकि
मैम।
ऐसा हो सकता है कि जब स्ट्रक्चरल एनालिसिस हो रहे हैं ना, उस केस में शायद हम नेटिव कंडीशन और ये सब चीजें होंगी उसमें। आई हैव टू चेक दिस थिंग, आई मे नॉट हैव द राइट आंसर, बट मुझे इतना जरूर पता है कि whenever we have our post translation modification studies done, we go for phosphoprotein enrichment, because that is what we study. First thing we study for post translation modification is phosphorylations.
ओके मैम।
Yeah.
मैम नेक्स्ट 10th क्वेश्चन।
Which of the following is not common for horizontal streak of two-dimensional gel? Overloading of okay. fine. So yes, when we do horizontal streak, समझ आती है आपको क्या है? What, which one is
Yes,
vertical?
ma'am.
When we do two dimensional, what is vertical and what is horizontal? First dimension is vertical or first dimension is horizontal?
Ma'am, when we are doing it on IPG strips, it's horizontal, and when we are running it on the resolution gel, it's vertical.
Okay. So you have written issue with when you are, which of the following is not a common cause of horizontal streak on a 2D gel? Not a common cause बताना है. तो जब हम वो कर रहे हैं, आइसोइलेक्ट्रिक चेंजेस कर रहे हैं, आइसोइलेक्ट्रिक के ऊपर हम बेसिस पे हम सेपरेट कर रहे हैं, तब क्या हमें डाइसल्फाइड बॉन्ड का फर्क पड़ेगा?
No, ma'am.
नो. तो आंसर क्या होगा यहाँ पे? देखो, ओवरलोडिंग कैन इफेक्ट योर जेल.
Hmm.
Uneven rehydration of IEG strip can affect your gel. Issue with pH gradient definitely going to affect your gel. बट क्या चीज फर्क नहीं पड़ेगी, क्योंकि यहाँ पर सेपरेशन हमारा
Hmm.
रिड्यूसिंग नहीं है, हमारा सेपरेशन सिर्फ आइसोइलेक्ट्रिक फोकसिंग पे है. तो यहाँ
Okay,
पर
ma'am.
पूछा है विच इज नॉट अ
Mm-hmm.
कॉमन कॉज, देन
Yes, ma'am.
डेफिनेटली इट्स अ डाइसल्फाइड बॉन्ड.
Okay. Okay,
गॉट द
ma'am.
आंसर?
Yes, ma'am. Yes, ma'am.
दैट्स इट.
Ma'am, I have some few more questions.
Okay, quickly, I just have other commitment after this, so
Okay.
I
Okay,
can't
ma'am. Okay,
take it
ma'am.
for a longer time.
Okay, ma'am. Ma'am, this one last question, sixth question.
So इसमें क्या बोल रहा है वो? In the system biology study, a researcher integrate transcriptomics and metabolomic data to analyze a metabolic pathway to find the expression level of several genes involved in glycolysis are elevated, but the metabolite level downstream of glycolysis are reduced. What could be the plausible explanation for this observation? बताओ. जीन डुप्लीकेशन. क्या हो रहा है यहाँ पे? प्रोसेस के अपर, मतलब अपस्ट्रीम सारे एलिवेटेड हैं एंड डाउनस्ट्रीम सारे रिड्यूस्ड हैं. दिस इज व्हाट इज हैपनिंग. जीन डिप्लीकेशन नहीं हो सकता. ट्रांसक्रिप्शनल रिप्रेशन. ट्रांसक्रिप्शनल रिप्रेशन में क्या होता है? दैट देर इज नो, रिप्रेशन के केस में क्या होता है? स्विच
मैम
ऑफ.
मैम या तो mRNA फॉर्मेशन नहीं होगा या तो डिग्रेडेशन होगा.
हाँ, तो ये तो नहीं हो रहा है, यहाँ पे प्रोसेस हो रहा है, मेटाबोलाइट लेवल रिड्यूस हो रहे हैं, अपर ग्लाइकोलिसिस पाथवे इंक्रीज हो रहा है. सो डेफिनेटली प्रोटीन तो बन रहा है, दैट मीन्स ट्रांसक्रिप्शनल लेवल पे चेंजेस नहीं हो रहे हैं.
ओके.
किस
ओके
लेवल
मैम.
पे चेंज हो रहे हैं? फीडबैक इनिबिशन जो हो रहा है ये. फीडबैक
यस मैम.
इनिबिशन इज, आपने ग्लाइकोलिसिस पढ़ा होगा. ग्लाइकोलिसिस
यस
में
मैम.
देयर इज अ फीडबैक इनिबिशन, एक एंजाइम काम करता है, एक एंजाइम
पीएफ
की
के.
प्रेजेंस में
यस
दूसरा
मैम.
एंजाइम काम नहीं कर रहा.
यस मैम.
दिस इज व्हाट इज हैपनिंग हियर.
ओके.
ठीक है.
ओके मैम. यस मैम. मैम, दैट्स ऑल.
Okay, thank you.
थैंक यू सो
All the
मच
best
मैम.
for your exam. Do well.
थैंक यू मैम.
Okay, fine. Thank you so much. Thank you for joining. All the best, everyone. I hope you give your paper well. If you have any other question, quickly ask. Otherwise, I'm ending this class. That's all?
यस मैम.
Okay, fine. Good night and all the best for your exam.
थैंक यू मैम.
Okay.